Thank you very much. So I was planning to talk about herrings today. If I get my slides, yes, here we are. So let me see. Okay, here we are. Okay, so I started with this picture, which is from one of the first books that was published in Sweden, 1555, and it's a book about all the history of the people in the Nordic countries and all the life there. It's a fantastic description of the life in Sweden and Scandinavia, I should say, in the 1500s. And one chapter is about the herring fishery. And this showed that at some points the herrings are so abundant that you could just stand on the shore and get all your fish there. And if you put it, sometimes the number of fish is so dense that you put an axe, the axe will stand in the water on the shoulders of the fish. But the fact is that, this is of course not true, but the fact is that the herring fishery has been enormously important for Northern Europe and the economic development of Northern Europe. Sweden would not have been a country without herring, so in the past, herring was the staple food during wintertime when nothing was growing. And there was the first export industry basically from Sweden. So the herring is definitely not a threatened species. It's a very abundant species. Could be one of the most abundant. It's one of the most abundant vertebrates on Earth. So a school of Atlantic herring could be 1 billion individuals, and they are present in this area of the Northern Atlantic. So both on the American side, Greenland, Iceland, around the British Islands. And something which is interesting for us is that we have the Baltic Sea here, so it's also into the Baltic Sea, which is a brackish water, which I'll come back to. So this is a product that I started more than 30 years ago when I was a student in Stockholm, student in biology. So I learned from fishery biologists that they have all these type of populations of herring. So Baltic herring is classified as a subspecies of the Atlantic herring. So you have the Baltic here, of course, the Baltic Sea, which is brackish. And then they have different stocks. You have North Sea herring, the pure pelagic herring, which migrates between Iceland and Norway. But no one really knows how genetically different they were. And I learned that you could start using isozymes. So this was before RFLPs, microsatellites, and any SNP markers. But you could use isozymes to study genetic variation in natural population. So this is a publication from, or a figure from my first publication where we see genetic polymorphism. So we decided to use this technology to study the differentiation of Atlantic herring and see how different are these different stocks or populations. Are they species, are they subspecies, or what are they? So I sampled 1,400 fish from 17 different localities all the way up here in the far north of the Baltic and along the coast. So it's a very big shift in the salinity. So the salinity up here is about 3 per mil, and then it decline, and then there's a big shift here. And on this side, you have marine water going up to 20, 25, and then out in the Atlantic is 35 per mil. So it's a big, important environmental variable there. So we found a lot of variation. We were able to study 13 genes. That was great at that time. But to some extent, the disappointing finding was that we found no difference at all. So all these certain loci show the same allele frequency, although we sampled in pure brackish environment. So this is the most extreme. So this is where the salinity is 3 per mil, and this is the Atlantic Ocean. And this is one of the polymorphic loci, GPI. And if you look at that, you see we saw the same alleles and essentially the same allele frequency. So what we could say was that the genotype distributions in these 17 samples was as if you have sampled one single panmictic population, just taking 17 different samples from the same population. So the question is, why is this? I mean, one is, of course, that there is no genetic differentiation. There's so much sufficient gene flow to hinder any genetic differentiation, and there are morphological differences, like the number of vertebrae, the size of but this could all be phenotypic plasticity. The alternative is, of course, that this colonization of the Baltic Sea is very recent. It's within the last 10,000 years. And if the effective population size, so only in the Baltic Sea, there are 20 billion Atlantic herring, so they have a potentially very huge effective population size. So if no genetic happens at selectively neutral loci, we need to really find those loci by scanning the whole genome. So since we didn't find any differentiation, I put this project aside until next generation sequencing was developed almost 30 years later of this. So the aim has been to use whole genome sequencing to see if we can find differentiation or not. So what we started out with was to make exome assembly. So we did a RNA sequencing from muscle tissue, developed transcript contigs, and those transcripts, we then took genomic reads and aligned that to the transcript. And so we made exome contigs. So here is an exon, and you have then the genome reads that map to this exon and then extend into the intron. And this gives us a better way to then utilize all the genome reads. So then you take whole genome sequencing, align to this contigs, do SNP calling, and then do statistical and phylogenetic analysis. So this is the experimental design that we took. So out of these 17 populations, we took 17. And we started up here, and we picked essentially all the representative populations throughout the distribution that we have access to. So this is a bit the history of the Baltic Sea. So of course, more than 10,000 years ago, there was glaciation, so there was no Baltic Sea. Then it became a brackish sea at one point. Then, so what is critical is how much flow of water there is here in this strait between Sweden and Denmark. So when this was blocked here, this became a freshwater lake. And then this, as the water was rising again, it's opened again, and it became brackish water. So you have rivers that feeds into the Baltic and dilute the marine water. So that's the reason why you have this decline in salinity. So in total, when we did this exome assembly, we were able to assemble 65 million base pair, which is about 10% of the genome. And when we aligned reads, so what I was supposed to say was that we took 50 fish from each of these 8 sites, and we pooled them into one sample, and then we sequenced them by pooled sequencing, every one to 30x coverage. And then we aligned them to the exome assembly, and we were now able to go from 13 loci to 440,000 SNPs. And out of those, it's a bit less than 4,000, but 1% of them show highly statistically significant differentiation between localities. So if you take all these 440,000 SNPs and make a phylogeny, you will see you get this star-like phylogeny, which looks like they are really similar at most loci. But there is, in fact, some structure in this. So what you could see here is that there is a little bit of structure here, and here you have the Baltic herrings. And whereas the Atlantic herrings goes into this group. But for most loci, out of these 440,000, it looks like this, like the one I showed you before, that if you take the allele frequency of 2 alleles, they are essentially the same across the range that we have sampled. If we instead just focus on these highly significant loci, this about 4,000, then you get much more resolution. And now you see the Baltic herring here. You see the Atlantic herring down at the bottom. So there is one interesting population, this one, which is a sample which is taken here, which is on the border between the Baltic Sea, which is here, and the Atlantic Ocean here. And in fact, there are some migration going on. So this particular sample was collected in October, so out of breeding season. So this guy, we can't exclude that this is a herring which has migrated out there, but will be spawning in the Baltic. So we need to study that in future work. So then it's a question, this differentiation, are we just looking at outliers, or is this drift, or is it selection? So that we did a simulation. So Nils Reimann was my supervisor as a student 30 years ago. So he was the one who kept the samples while I was doing work on domestic animals and other things. And then we decided to collaborate, and he did this simulation. So what he did was to simulate genetic drift. If you split up populations and allow them to drift to give the same average FST value as we had in the observed data. And then we compared the simulated data with the observed data. And what you could see on this slide is that there's a very good correspondence between the simulated data and the observed data, except for this here, which is a tail of high FST values. So what happens is that you have more, so you have FST on this axis. So in general, most loci have close to zero FST, but you have some loci that show higher FST than you expect by drift only. So this is, if you look at that tail of FST values, so these are the simulated data, so you don't expect to find more than 15% or FST of 0.15 or something like that. Whereas you have this tail of loci that really indicate that. So our conclusion is that 2 to 3% of the loci show more genetic differentiation than expected on the basis of neutral genetic drift model. So that we consider this evidence for natural selection. So as I said, we made this allele frequency by sequencing pool samples, and these were 30-year-old samples, so we wanted to verify that the loci that we identified as outliers was true. So we made a chip panel, so 5,000 SNPs, so all the highly significant SNPs that we identified and a similar number of non-significant SNPs, and we got data for about 3,000 of those, which were given reliable, high-quality SNP data. So if you take the non-significant SNPs, or control SNPs, and make a phylogeny, so this is what it looked like. It's exactly as we saw before. We don't find any differentiation, so there's no structure if you take random loci. When you do, so now we have typed in the same individuals as used in the pool sequencing, but now genotyping them individually with the SNP markers. If you instead take these markers showing differentiation, then you get a nice. So here we use a software called Find Structure, which try to find the relationship among individuals without having any previous idea about what is the relationship. And then you could see that the Baltic herring cluster quite nicely here. Here you have the Atlantic herring on this side. And here we have this population, the one from Kattegat, which was down here, as I mentioned, which tend to cluster with the Baltic herring, and that's something we need. And you could see also that in most cases, all fish from the same locality cluster together. So you could see here is, this is from the Skagerrak, here is from the Atlantic Ocean. These are the ones which is sampled out here. You have the North Sea, I think is this light blue. It's difficult to see here from the side, but overall it gave a very clear indication of genetic differentiation between the different populations. So this is something that if you take one example, so this is one of the SNPs showing genetic differentiation. So you have Atlantic herring starting Atlantic Ocean. So you start out here, and then you go all the way up to here, and then you could see that you have this nice climb of in allele frequency. So you go from about 10% to almost fixation when you're out in Atlantic or in the North Sea. And in this case, it's a gene called, so the advantage with this exome assembly is that all the SNPs we have is linked to a transcript that we could search against other species. So this is a gene called glutamine synthetase, which is associated with adaptation to salinity in several fish species, based on physiological experiments, not genetic experiments, but we have a strong genetic signal at that locus. So this is another thing we could also, so we have these 4,000 SNPs showing differentiation. So how are they distributed in the genome? And what you could see, so at this stage we didn't have an assembly of the herring genome. So we use a stickleback as a proxy because there's a lot of conserved synteny between different fish species. So what you could see here is that this loci that show is expected to be distributed across the genome to a large extent. But there are some clusters. So you have here, you have clusters of SNPs. And this is one example, because when we found these SNPs with very strong differentiation, we asked what genes are they? So we went to the zebrafish genome and asked what is known about these genes and so on. And then we realized that a number of these were in fact sitting together in the zebrafish genome. So here you have an example where we take SNPs and we take the order of genes on this. This is the stickleback genome. And what you could see here is there's a very, very strong correlation in allele frequency So out here you have the Atlantic, the North Sea, and one more of these populations. Here you have essentially the Baltic herrings and this Kattegat sample, and this is a population that is present between the Atlantic and the Baltic, so in this area which are between the open Atlantic and the Baltic Sea. And our suspicion here is that we may have an inversion that is explaining the very strong LD among these. So you get the same, and that means that SNPs that are diagnostic for that inversion will have the same allele frequency. And that's why you have such a remarkable correlation in allele frequency over a fairly large region. So even in this case, we have one good candidate. So we have a gene which is involved in the transport of a calcium sensing receptor that is involved in salinity sensing. So this is another example of what we can do. We could query this data. So here we show us some of the loci with the most striking difference between the Baltic herring and the Atlantic herring. And for some of these, there is like a perfect correlation. So you could take this one as an example. So you have dark red means complete fixation from one allele, whereas white means fixation for the other allele. So you have here you see one which has like a perfect correlation with salinity. Here. And if you look what that gene is, it's a vacuolar ATPase proton pump involved in osmoregulation. So what I wanted to say then is the ongoing work we're doing at the moment. So now we have draft genome assembly. So we have worked together with BGI to generate an assembly. So to generate a good assembly, the first experiment to use the exome assembly was basically to see, are there any differentiation, or anything to be learned about the genetic differentiation in this case? Because if there have been no differentiation, then we didn't need to go for a whole genome assembly. But now we think we got the motivation to do that. So to generate a good assembly, you need to have different type of libraries. So this is pair N libraries. And then you have a number of mate pair libraries. That the problem, of course, to make an assembly is to bridge regions of highly repetitive sequences. Otherwise, your assembly will be very, very fragmented. And for that, to bridge those repetitive regions, you need mate pair libraries of different sizes. So that is what has been generated. So in total, we have generated 120x coverage of the herring genome with different library sizes, and this has resulted in a good assembly. So this is some details. So the estimated genome size is 700 megabases, and the assembly we have is in fact a bit larger, and that's because you put in ends where you have this, where you can't really bridge over. But overall, what we could say is that if you look at the scaffolds of this assembly, so if you more than 50% of the genome is in scaffolds that is 1.7 megabases or larger. So it's a very promising assembly that we hope to improve in the near future as well. So to have a genome assembly is, of course, make a huge difference. So when we now screen the same samples, because now we could reuse this genome sequencing that we did before. So now the number of SNPs goes from 400,000 to 8 million instead. And what is the really major advantage here is now we can order them along the chromosome. We don't need to rely on the association to the stickleback genome or the zebrafish genome. So now we could order them perfectly, and we could have coverage across genes and intergenic regions and so on. So that allows us to define the regions under selection and their gene content to get greater resolution. And we could, of course, do a comprehensive search for sequence polymorphism in coding and non-coding regions and study the pattern of variation. So another thing that we would like that we are doing is to do resampling. So the first sampling I did was 1978 to 1980. So the second sampling have been going on 2011 to 2013. And there's been a considerable environmental change during this period, as you know, as you heard during this conference at least. And warmer winters, you have less ice in the Baltic Sea, earlier spring, so spawning could start earlier. There's also more intensive industrial fishing now. And the herring, as I said, in the old day, the herring was used for the people in the north to survive winters. Nowadays, herring is to a large extent used to make fish meal to feed salmon, which is we export to Norway, where they make salmon, and then that's exported back to Sweden and to other countries to have fresh salmon. And there's also have been intensive fishing on the cod. So cod is one of the major predators of the herring, and the cod has crashed in the Baltic, for instance. So there's a change of the ecosystem that could have impacted these populations. Something we also, so in the Atlantic herring you have spring spawners, summer spawners, winter spawners, and autumn spawners, and so we have sampled the same locality where they are spawning, but at different time periods. And the question is, of course, is if this variation in spawning time is just due to phenotypic plasticity, that they could shift when they are spawning, depending on how well fed they are, or whether it's a genetic adaptation. So what I think is really, I think the herring spawning is a little bit what you heard about yesterday about the flycatcher breeding. So the flycatcher need to breed to hit the peak in the preserved caterpillar. The herring need to do the same thing, because when their fry hatch, they need to hit a stream of water where there's plenty of zooplankton which they could feed on and grow. So we're looking forward to analyze that as well. And then we do also an increased taxonomic representation. We have included Icelandic herring now as well, and the Pacific herring, which is another species. But the Atlantic and Pacific herring could meet, in fact, in the sea north of the polar sea, so to speak. So there you have Atlantic and Pacific herring. So we would like to see if there has been gene flow between the two species and how divergent they are. What we could also do with the Atlantic herring is to do breeding experiments. So that is what we did this year. So we had, in collaboration with a group in Bergen in Norway, we have crossed the Baltic herring and to itself, and with Atlantic herring, and Atlantic herring to itself. And then we have raised them at different salinities, because this is obviously one of the environment factors that the herring is adapting to. So what we will do from this experiment, so we sampled fish this week, in fact, earlier this week. So we will then study how different genotypes can handle deviations from their normal salinity. So that is to measure phenotypic plasticity. We will do RNA-seq analysis to study differential gene expression, which is when you compare gene expression in different environmental conditions, the salinity in this case, difference depending on the genotype, and look for genotype environmental interaction. And we also plan to look at DNA methylation. So will you see changes in the methylation patterns that then alter the expression of genes when you expose the animals to different salinities? So that is another thing which we plan to analyze in this material. So with that, just a few conclusions. So we think this is a breakthrough as regard the characterization of the population structure of marine fish. Has been huge important both for fishery but also ecological importance, because it's feeding on zooplankton, but it's a feed for birds, whales, other fish. And we think it provides an excellent opportunity to study the effects on natural selection when you don't have, when the effect of genetic drift seems to be so mute as what we could see. So that is what I think is a strength with this system. And I think what I showed you with this exome assembly is an extremely cost-effective way to get a quick overview of the population structure of a species like this. So that's what I end by acknowledging people which have contributed to this study. So this is from my own group in Uppsala. So Alvaro Martinez Barrio had done a lot of this, and he's also present at this conference. SciLife Lab Uppsala has been doing sequencing, and the Beijing Genome Institute has been involved in sequencing these mate pair libraries and doing the assembly using their Soup de Novo software. So RL Folkward is doing working with these common garden experiments and breeding experiments in the herring. Our ambition is, in fact, to keep this herring until they are sufficiently old to make F2 generation, so we could do genetic segregation analysis. And then Niels Riemann and Linda Laikre, so Niels particularly for keeping this fish for 30 years. Thank you very much.